And let's move on to the next presenter. And the title is Smart Drug Delivery Platform based on Cell-derived Vesicles, and which will be delivered by Dr. Seung, and he serves as a CSO from MDimune, and also he serves as CEO for Biotherapeutics, and he also earned his Ph.D. from University of Massachusetts, and also Dr. O has been leading the development of a clinical trial and project at Allen Institute. Please give him a big round of applause. Thank you very much. Good morning, ladies and gentlemen. As previous speakers are talking about the commercialization of exosome-based therapeutics, and it's my great honor to be here with these eminent leaders. And they talked about the reason why exosome is getting great attention, so that's also makes it easy for me to deliver my presentation. And exosome and vesicles are utilized in the talked in previous presentation. And we try to come up with new strategy to commercialize these exosome-based therapeutics. So actually, how can we commercialize those idea of exosome? And actually, that originated back to 10 years ago. And actually, my next speaker, Professor Ko from Postdoc, actually developed extrusion-based vesicle methods, and we got technology transfer. And in 2015, our moderator established our company, and we have employed more than 40 global employees and 13 research personnel, and our office is Seong in Seoul. And in Seattle, we have global office focusing global BD. And CDV is a new technology, and GMP production process development are done by experienced veterans in exosome manufacturing. So exosome manufacturing pioneer Mario Zimona, doctor and director of Berkeley Medical University, and we have enjoying joint partnership, thereby laying foundation for commercializing our products. So I can skip this presentation because you're all aware of, and you are all aware of the reason why exosome have come in spotlight. And the exosome researchers and in 2014 and 2015, the attempts to commercialize the exosome therapeutics have occurred. And in the early stage, many companies and research institutes think that, and the benefits of a base approach are natural messenger in cell-to-cell communication, and they are focusing on development of EV. But at the same time, they try to increase yield and manufacturing efficiency. And this is part that I would like to elaborate more. And so there is one way, extrusion-based method. And with this method, exosome potentials can be realized into a commercial product. And we are doing that exclusively. And actually for 2 to 3 years, US Kodiak, the UK Evox, and they are pioneers in the exosome field. And they have collaboration with large big pharma. And actually they have concluded a deal which is amount to $1 trillion. Actually, this is not in clinical trial, this is in preclinical trial, and this large deal was agreed, which means this is a clear testament to how much attention to be maintained in the global big pharma companies. So now let me take a deep dive into our core technology. As I mentioned before, one of the issues here in researching exosome is about exosome yield. So exosome can be naturally secreted by cells. So cell status as cell type and differentiation of cell and the culture environment are factors that affect and decide exosome yield, and artificial adjustment has limitations. So going beyond that, so we think about how to easily obtain exosomes and vesicles from cell. So that got me develop extrusion technology, and the principles will be elaborated in this video. And cells goes through physically stressful environment, and conventionally we use membrane filters. So cells should go through a small hole, and there are a lot of pressure which leads to disrupt of cells naturally. And to our surprise, this broken down membrane naturally form a ball. So a variety of extrusions leads to miniaturized cell vesicles. So a lot of research support this, and these vesicles are similar to natural-driven exosome in terms of physical property and functionality, and CDV-based technologies are well known to this. And so we manufacture vesicle from extrusion, and cell engineering, vesicle engineering are used to load biological cargo and chemical cargo in this vesicle. At the same time, surface around it can be engineered in a variety manner so that we can engineer a more efficient vesicles that is not obtainable from naturally driven cell vesicle. And also in the early phase, 1 ml syringe-based extrusion was used. So it's a small syringe for your understanding. So we attach filter on the syringe, on top of the syringe, and we pull cell into this syringe, and it leads to extrusion of cells. And in the early stage of our establishment, we have been focused on how to scale up this at an industrial level. And with years-long researches in 2019, 100 milliliter cell suspension was intruded at once. We call this intermediate extrusion, and also intermediate scale extruder are allowing us to produce a larger scale of vesicles. And also, in order for these to be commercialized, there needs another step of scaling up. So last year, larger scale refined process and extrusion for research for that are under development. And now we are able to extrude 50 liter at once, and we have developed prototype for that. And to be commercialized, we have cooperation with manufacturers and other experts as well. So there are applications of our technology. For your understanding, there are two parts for here. So on the left side, and on the left side and this, you can see regenerative medicine. And there are a lot of cells that are very suitable for therapeutics, such as stem cells and other immune cells as well. And our vesicle technology ultimately breaks down cells, and we try to produce cell-like or miniaturized cells that are very similar to cells. And also the when the mother cell is vesicularized, those characteristics are inherited. CDV researchers and us are already proving this. So building on that, let's take stem cell as an example. So when we obtain vesicles from stem cells, it allows us to overcome the tissue barriers so that the therapeutic functionality of stem cell can be delivered to target region. And also in terms of immune oncology areas are focusing on a lot of applications. On the right side, actually on the left side lies stem cells and immune cell, and it is the most weak point of primary cells are the difficulty of being engineered. So external genetics and materials in section are having limitation here. So in order for us to advance vesicles in terms of engineering, it also lies a challenges. So to overcome these challenges, we have utilized a lot of methodologies to hack cell to produce vesicles, and produced vesicles are used to obtain vesicles with high functionality that we are not able to obtain from primary cells. And through this, we try to load cargo to be delivered to the target region. So we try to develop this as a smart drug delivery platform.
Let me tell you about the strength of the vesicle in 3 ways. First, our technology itself is trying to overcome the limitations of the naturally oriented vesicles. So the first benefit is scalability. I need to mention this. When we talk that this is scalable, of course, we have developed many processes for scalability, and already there are studies in the clinical phases. But when it comes to the stem cells, when we use the conventional purification process, we can obtain about 1,000 exosomes. However, from one cell, now we can obtain 30,000 CBB particles, and more than 200 times of yield gaps can be observed. So this differentiated productivity can lead to an easier operation of the process, and we can also reduce the cost. So we can create many benefits. And the second point is, as mentioned by previous speakers, this is oriented from human cells. And compared to synthetic vesicles, viruses, or cells, these vesicles we developed can be more safely delivered to human body. And another characteristics is the versatility. It can be viewed in many perspectives, but the first thing I would like to emphasize is that there are some cells that does not create exosome easily. In that case, even though we have good elements in the cells, but there are limitations in commercialization of the vesicles. But with the physical ways, we are making smaller vesicles. So it is not a natural way, rather it is based on the physical characteristics of the cells. So regardless of the cell culture condition, we can produce the vesicles. In this case, I believe we can expand this. And in exosome field, we are studying various technologies, and these technologies can be easily applied to our technology. So that's why I can say that our technology is very versatile. For the sake of time, I will only mention some of the data that can show the characteristics of the CDVs. When you look at this data, it is difficult to differentiate, but the blue one is a CDB and the red one is exosome. And the top one is exosome, and bottom one is the vesicles. So with extrusion, when we create the vesicle, physically it is difficult to tell the difference. And once the vesicle goes through enough extrusion, compared to the naturally made exosome, the physical characteristics and profiles are almost similar to the original cells. And in the middle, you can see the 3D structure by using the imaging technique called the SEM, and we can see that the particles are very consistent. And another characteristics is when we develop the CDV technology, eventually we want to deliver the drugs, and we would like to deliver the therapeutic molecule inside the cells to the cells. So the characteristics, of course, delivery should be considered first, and the cargoes should be released into the cells. But first, when we look at the uptake of the cells, we can observe repeated behavior. This is one of the data. We received the extruded vesicle and naturally obtained vesicle, and after purification, we used the vesicles, and from the extruded vesicle, we can see higher uptake in the cells. So these red dots are vesicles, and at the bottom you can see the exosome. We also used 4 types of cells, but we can observe the similar situation in other cells as well. And in the CDVs, the uptake was higher than naturally originated vesicles. And we also had an injection, and the efficiency was higher in the eyes. So a CDV can go through the retinal barrier, and it can also target many retinal-related diseases. And stem cells and other materials are now being developed for a cure for retinal diseases, but using the vesicles, we can easily and effectively deliver the therapeutic effect. So this is a case where we can showcase the benefits of vesicles. And various mechanic studies have been carried out, and we have the clues that proteins make this difference. We created these vesicles, and in order to go to the next step, we need to have the GMP level manufacturing process. And for the last 2 years in Australia with the PMU, we collaborated with GMP facility. And as I explained before, we are using intermediate scale extruder to develop GMP compliant process, and it has been completed already. And documentation and process has been established. And the vesicle produced here will be used to understand the characteristics. And the stem cell CDV may have a lot of therapeutic effect, especially anti-inflammatory effect. So these CDVs are listed up here, and in various diseases, we can see the in vivo effect when it comes to the anti-inflammation. Due to the limited time, I will be brief. There are many ways to load the cargo, and these good technologies should be applied to our CDV. Of course, because the vesicles have the similar chemical and physical characteristics. And so small RNA and messenger RNA, these large cargoes can be loaded in various ways so that we can see the effective outcome in the in vitro situation. And proteins are also being studied. Last but not least, in our vesicle, with additional engineering, now we can use the therapeutic antibodies, and we are targeting the tumor and to go through the BBB. In other words, we are now developing selective vesicles. We are developing we are not developing the therapeutic pipelines, but eventually we can support therapeutic pipelines. So we are developing good platform technologies, and through various partnership, in the future we will support more pipelines. With this, I would like to conclude the presentation. Thank you very much. Thank you very much.